Welcome to the Women Inspire Aspire podcast. In this episode, I talked to Kinjal Sanghrajka about brain cell functions. Hi Kinjal, thank you for accepting my invite to appear in the podcast. How are you doing today?
I'm doing good. Thank you for inviting me to this podcast.
Wonderful. It's a pleasure to have you here. Can you go ahead and tell a bit about yourself, where you are from, where do you live, etc.?
I am Kinjal Sanghrajka. I am from Mumbai. I'm born and brought up here. I did my schooling and bachelor's from Mumbai. I went to Baroda for a short 2 years master's program and then I came back to Mumbai. Just finished my Ph.D. degree from TIFR Institute, which is Tata Institute of Fundamental Research from Mumbai.
That's wonderful. So what is your research about? Objective of the research and how it is helping the community?
My basic research at TIFR is if you understand a brain communicating day-to-day activities, it requires a brain cell communicating with other brain cells. So my entire project dealt about how neurotransmitters or the chemical messengers are released from one brain cell and understanding what is the structure and function and the dynamics of the protein which carry out this essential fundamental process of communicating between the two brain cells.
Okay, great. Before we go into the details of the work that you are doing, let's look a bit back into your childhood and what made you pursue the field of research, especially the science research.
I took up science after 10th standard. Back in those days, it was like once you had good scores, people would encourage you to take science, and my parents did the same. But I was always in the mindset that I don't want to do a typical doctor or MBBS or engineering from the beginning. I had undertaken biotechnology for my BSc, and that time biotechnology as a field was not very famous. Took up biotechnology, and in my undergraduate program over there, I was introduced to research for the first time, where our HOD encouraged us to do very small research of isolate bacteria from cell, and that is how I came to know that what research is actually like. Thankful to my master's degree in Baroda at MSU University, we had a 1.5 year research project where you are given an independent project under a guide. My project was also dealt with microorganisms. That 1.5 year of rigorous training to find out experiments, sometimes experiments fail, hypotheses fail. That rigorous training of 1.5 years actually pursued or made me more strong that I want to do research which will find out something new.
Okay, so how did you select this particular line of protein and brain cells related?
I was always interested in doing neuroscience as a field. I was curious as to how people understand the brain itself, brain being the complex organ. When I was introduced to this lab, so this protein, snare protein, which actually carries out the fusion of two membranes or first step which is required for the brain communication, these proteins were discovered like in 1990s. So it's almost like 3 decades since these proteins are discovered. We know the broad end picture that from one brain cell to the other brain cell, the communication is happening. But we still don't know the molecular details of it. And because of this, there are a lot of neurodegenerative diseases like Alzheimer's, Parkinson's. Your brain communication has gone wrong. But if we don't know the detail of how this actual process is happening, it is very difficult to target personalized drugs which can counter this problem. So this actually really fascinated me that we know all the broader terms of it, but we still lack fundamental details of how things actually happen inside your body, and that intrigued me a lot, and that is how I ended up joining this lab and pursuing minute detail as my PhD topic.
Okay, great.
If you consider any normal signaling, you should receive an external signal. So let's say if you have a brain cell, if you want to move your hand from one place to another, your brain receives an input. Okay, that this is the input that I need to move this thing. And then it relays that signal to the next signal as an output signal. Imagine that this is all happening in a milliseconds to microseconds time scale, because your brain communication happens at that time scale. In our brain cells, there are small tiny packages called vesicles which have the neurotransmitters or the chemical signal required for the transfer of this fusion. They are all kept ready near the end of the cell or the plasma membrane, and it is all kept ready over there. Once the signal is received, you have some changes in the calcium ions, and that is how the tiny package fuses to the plasma membrane or the outermost membrane, and the package package is now released. Now these proteins which I'm working on, the snare proteins, are the ones which gives the energy to this entire process. So this undergoes a series of interactions and releases this protein. Now this entire process has to be tightly regulated because you don't want signals going randomly to all places at all times. So it should be timely regulated as well as spatially regulated. For example, if I want to move my hand, my signal should go from my brain to my hand at that particular time only and not to any other organ. So that is how it is very tightly regulated. So my entire basic project dealt about, if you have so many regulators, so many positions, how do you spatially and temporally regulate it via the same protein? So I used to look into connections within the protein, that how is it differing in the terms of structure, in terms of function, in terms of dynamics, to incorporate all the major changes which can happen. Generally in diseases like Alzheimer's or Parkinson's or any neurodegenerative diseases, it starts with signal going wrong. Either it is sending too much signal to one particular cell, or it is not sending signals to that one particular cell, and eventually the neuron dies off because of aggregation of other different proteins, loss of signal, and that path eventually closes, and that leads to the broader symptoms that one sees in a person suffering from neurodegenerative diseases.
Okay, that sounds like a great description of what you're doing, and it's very explanative. So thank you so much for sharing that. As the end of your research, as a result of your research, what are the advances that you made from where you started and where you ended your PhD? What are the results that you got out from your work?
Did find out that this protein basically has three different parts. So the protein itself is divided into like three different regions. And we found out that within these three different regions, it has a set of communication within that protein. So let's say an X protein, which is supposed to start the reaction, binds to it, a different connection is formed within the protein. And when a Y protein binds to it, which has some other function as opposed to the previous one, it forms a different connection. So basically the protein knows that what kind of regulator comes and binds to it and what kind of downstream action it has to take. So that was interesting, and it is relevant also because you have like hundreds of different regulators which either will stop that reaction or start that reaction, and it all binds to one particular region only. Now how will the cell or how will the proteins know that what kind of reaction it is? Therefore it is very much interesting how these two interactions happen, and this is what we found, that certain kind of reactions, certain kind of regulators activate one pathway and certain kind of other regulators activate the other pathway. This was the gist of understanding of my entire project, that the protein itself has one intrinsic regulation on top of all the other regulations which happen inside a brain cell.
Okay. With this knowledge, how do you think the next set of researchers can take it forward?
Basically, right now, the field is moving towards lot of personalized drugs or peptide-based drugs, which can have direct targeted effect towards the action, towards your affected region, and not have a haphazard side effects of all these drugs. The reactions that performed in the lab, it was all chemical reactions. It is not inside a brain cell. What we did is we identified specific regions which are responsible for one set of experiment or one set of pathway inside a brain cell. Now, if you can design peptides which particularly stabilize that sort of interaction or destabilize that sort of interaction, then one can control that pathway by smaller peptide drugs, and this can be exploited as a drug delivery system or a drug target in future for other neurodegenerative diseases as well. So this is how I look forward to the pathway of this project. Of course, it requires a lot of additional studies as well, but this is one angle which can be exploited for the benefit of the humankind.
Every body is different and every situation is different in terms of the diseases that people are getting. So how can you design medicines which are generic, which can be applied towards, multiple individuals? It might not work, right?
It will not work as a generic medicine, but in cases of neurodegenerative diseases, it's most of the time accumulation of lot of mutations. There are widely mapped mutations which is considered as a starting point of this disease, or which is most accumulated in the disease patients also. So if we can identify those cohorts where this is the maximum repetition of this particular change, then one can design peptides towards it and then help cure the disease or maybe undo it to a certain range. Not completely reverse it, but maybe help it stabilize it or not go into a lot of degradation.
Okay, wonderful. Now, one curious question that I have is you have done this study, right? How come this not been done in the past? Why now? This has not been attempted in the past, the study of the protein?
So protein has been studied widely, but because there are caveats to the existing experimental setups that one has, because one thing is the brain communication happens at milliseconds to microseconds time scale. Capturing anything at that speed is limited by the experimental setups which exist in the scientific community. Second thing is we know the proteins, we know a lot of regulators, but we still don't know the structure of these proteins. So if you imagine there is a hole or a pore which is being formed between the two membranes or between the two lipid entities through which it's a passage which is formed through which these neurotransmitters or the chemicals are being released. Now that structure is not resolved. So we are limited by structural studies, we are limited by functional studies as to how something can be targeted at this fast time scale. And that is why people face problems in delivering certain drug targets at that particular stages. So most of the studies which happen for the neurodegenerative diseases, the major drawback is that there are no early onset detectors for it. It is always when you see visible symptoms in a person where you can detect the disease. So major field is now moving towards finding out early detection systems as well as early diagnosis or early treatment diseases by which you can maybe halt the disease or make it a slow progression as compared to the fast progressive disease.
Okay. Now we talked a lot about your work. Now let's talk about your hobbies and interests. What do you do for fun apart from work?
So apart from work, I really like to paint. So even in my hostel, I used to have a set of painting colors and I used to paint over there. TIFR has a very nice sea face also. So love watching sunsets. So that was also one of the hobby. And I used to like cooking. And so cooking, traveling and painting are like my hobbies. But cooking, I could not pursue it much when I was doing my PhD. But apart from that, traveling, watching sunsets and painting was my most of the favorite pastime when I'm not working.
Okay, so what are the places have you traveled so far?
Within India, I have traveled to the north, Manali, traveled to Rajasthan. I have traveled to a little bit of south in Chennai. And apart from in India, outside India, I've been to Europe. I have been to Europe twice and then I've been to Bangkok and I'm still open to exploring new places.
Okay, wonderful. Now let's talk about the societal issues that you care about. Of course, you're working on research and that's contributing a lot to the society. Apart from that, are there any societal issues that you care about?
I am actually interested in making awareness, spreading awareness about research in general and what all career options one can have when you are very early at age. Because research in India is not very well pursued as a career option. I personally was not exposed to research until my undergraduate or a postgraduate when I was doing my master's. So at TIFR, we used to host a lot of outreach experiments where we called school kids from different places around Mumbai and we used to show them small scientific experiments and have a brief exposure about what research is like. As a person in the scientific community, I would like that people from all different places, not just about research, but should at least be aware of what all career options or what all options are available in the country where you can pursue apart from the mainstream career options like pursuing a CA, pursuing doctor, pursuing engineering, which have been mainstream for a lot of times. Right now, a lot of fields are evolving, but most of the people are not privileged enough to know that information and choose for themselves that what career options do they like. So I would personally want more exposure to school kids and also I like spreading education or spreading knowledge around the curious mind. So I like teaching as well. to make them aware about the different options that we have.
So how are you planning to go about it? Are you planning to start an organization to do that? Are you planning to consult in the schools?
I plan to do it via these outreach programs only, because now the initial steps that I can take is PIFR had a very good outreach program. It was a good platform for us to go and visit school kids, and later on, maybe if I get some NGOs or some schools which are ready to conduct workshops like this, even in science there are different fields which can be explored to do your PhD, and they have those sort of workshops, and I would like maybe try that, make people aware about it.
Okay, now that you've completed PhD, right? So what are your plans?
So I am planning to join IIT Bombay as a postdoctoral fellow. So I am currently in application process. I've sort of identified a lab which will host me for the postdoctoral fellow. So I will soon join that lab as a postdoctoral fellow.
Okay, so we are coming towards the end of the podcast. Do you have any questions for me or about the podcast?
No, I would just like to share my thought that it's great that you are having this podcast with lot of females in general from our country, and what they are doing in life to actually inspire a lot of people around us that yes, if someone else can do it, we can also do it. So thank you for doing it.
Okay, thank you so much for appearing the podcast and sharing your views. I wish
Thank
you
you.
all the best in your life and whatever you aspire to do. Please take care and bye for now.
Bye. Thank you.
That was a great podcast with Kinjal Sanghrajka. Please subscribe to this channel and follow it for more awesome podcasts. Opinions expressed by the guests in this podcast are their own. The host or the sponsors of the podcast do not condone the opinions expressed by the guests or the actions arising out of those opinions. Any decisions or actions that the listeners might take as a result of listening to this podcast must be based on their own judgment and will be at their own risk. The podcast host or their sponsors will not be held responsible for the consequences of any actions and or decisions taken as a result of any content in this podcast.